last topic of conversation, which is clean rooms. And Kevin, you raised a point earlier when we were talking offline about the need for a clean room in peptide synthesis versus when not. Specifically, when exactly does a synthetic peptide need to be manufactured in a clean room versus when can it be made in a standard facility before moving to a clean room after a critical synthesis step, as well as specific clean room classifications for peptide manufacturing. So, Kevin, would you mind weighing in on that?
Yeah, this is an interesting debate because it kind of comes about as part of the in the United States is designating BLA versus NDA being somewhat arbitrary, 40 amino acids in length, and the resultant requirements for room specifications for those syntheses. And I think it's important that we actually get some clarity around when exactly a classified room is required if we're doing basically peptide synthesis or chemical synthesis. Is it necessary to really have the type of room classification that's really more important in the recombinant space where contamination is really a challenge? And at what point do we draw the line that says, well, a qualified space really doesn't have to be a part of the manufacturing infrastructure until, say, the purification of the crude to the pure? And I think some guidance in that space would be really helpful because, the arbitrary nature of saying, well, at 40 amino acids, now it's got to be in a clean room versus when it was 39, it could be in a more traditional synthetic facility, is difficult to navigate. And so it would be great if we could get a clear understanding that synthesis, pure chemical synthesis, can be run in a more traditional facility as long as that designation for the room classification doesn't occur until we're really getting into the final asset, the crude to pure steps.
Great. Thank you, Kevin.
Which, by the way, is the same, what Kevin mentioned is the same approach that is taken for small molecule, even if the small molecule is injectable. Until you arrive to the final isolation step, you don't need to be in a clean room, to the point where you have to do the last step of the process, the crystallization and the drying, probably in clean room. But even sometimes, if you're working in a closed environment, in a closed equipment, you can do all the discharging, just the discharging in the clean room if you have a clarification of the material. One of the challenge of peptide, most of the peptides are injectable, so they have to have a certain level of cleanness at the end of the process. But this is the same thing that happened for something like, I don't know, propofol, which is, I don't know why I'm thinking of something that visually I know because I've been injected propofol for some operation. And this is an injectable liquid molecule that is the end of the process is conducted in a clean area because it's injectable. But there are many other injectable small molecules in the same approach. So Kevin is absolutely right. We need to define when it's necessary, but we need to keep in mind that the peptides are injectable. For 90% of the peptides are injectable.
Yeah, I think most of the concerns associated with biological processes versus small molecules, including peptides, has to do with risk associated with contamination, and the risks in chemistry-based processes where there's a significant amount of solvent are just intrinsically low, and so for bacterial contamination versus for biological processes where much higher, they tend to be aqueous, neutral pH, those kinds of things that isoosmotic, the things that potentially increase the potential for growth. And so I think that's why historically the small molecule world for sure, and the peptide world for the most part, low risk, really, for contamination. It isn't like, as Matteo said, it is an injectable product. So at some point, you want to introduce that control, probably use higher quality water, those kinds of things in the final step, just to ensure that you check some boxes associated with contamination.